In this unit, we cover animal diversity. We won't specifically be talking too much about aquatic versus land. We will talk about some of the important characteristics that allowed organisms to move onto land. But generally, we are going to start with our invertebrates, who tend to be, especially our earlier organisms, aquatic. And then as we move forward and have a variety of new characteristics, we start to see organisms move on to land. And then, of course, our animals are a huge group of diverse organisms. We'll spend one chapter on general characteristics of animals altogether and invertebrates. And then in our next chapter, we will talk about the vertebrate groups or the chordates. Depending on the semester, if we have time, we'll do some lectures on animal tissues. We may have already done some plant tissues. We'll definitely do a lab that talks about some of the different types of plant and animal tissues that we see. So some of this will overlap with what you've learned in lab. Some of the types of characteristics are going to be similar to adaptations we see in plants. And you'll notice that there's also a lot of vocabulary in the beginning of this chapter, a lot of new material that talks about the structure and function of animals. We haven't done a lot of that yet. You may have had that introduction from the tissue lab, depending on the semester, but otherwise we're going to have some new characteristics that we didn't really talk about in plants and protists because they're so unique. So just a couple of things. Keep in mind that all our animals are going to be multicellular. We are in the Uniconta supergroup of our eukaryotic organisms, specifically the opisthokonts. So because we are in our protists, we are also, all of these organisms are going to be eukaryotic. And we are in one of the three phyla where we see a whole bunch of multicellularity. We see multicellularity in those plants, in fungi, and in animals. Because they're animals, they lack cell walls, and they also are not performing photosynthesis. So this is probably not a surprise to you, but that does distinguish it from our plants, and then cell walls distinguish it from plants and fungi. So we're going to start this chapter by talking about some broad characteristics of animals, kind of some general classifications, just like we did with our protists, kind of some general broad categories, not necessarily monophyletic categories, more like descriptions of these different organisms. So at this point in the semester, you are well aware that animals are heterotrophs. You probably knew that already. Animals ingest their food. So they're not performing photosynthesis. They must get their energy-containing molecules from other living organisms. So this could mean from plants if they are herbivores or omnivores. It could mean other animals if they are omnivores or carnivores, or some of our more specialized groups like the detritivores. But they are all going to be ingesting their food. The major types of feeding strategies include suspension feeding, which is filtering food from the water. So some of the organisms like our jellyfish, our sea anemones, some of those where they're sort of sessile or just don't have a developed mouth, they're going to be pulling in organisms right out of the water. All of their nutrients are going to be essentially dissolved or suspended in the water. Then we've got bulk feeders. You and I are bulk feeders. This means we eat large pieces of other organisms. In other words, we take bites and we generally chew them up, although not everybody has teeth. Then we've also got fluid feeders. This is not the same thing as suspension feeding. Suspension feeding is filtering the water. Fluid feeding is sucking out the fluids from another organism. So for example, organisms might suck out the sap in plants, which would come out of the phloem, the sugary syrup-like liquid inside of our plants, and then animal body fluids, excuse me. So for example, mosquitoes will poke their proboscis through the skin and draw out blood. All of our organisms, with one exception, are going to be capable of movement. And that one exception is going to be our sponges. We'll talk about those when we get there. But everybody else is going to be capable of locomotion. In order to do this, you generally need both muscle and nervous tissue, especially for coordinated movement like we see in our higher animals, reptiles, mammals, fish, things like that. But even our earlier diverging organisms are capable of some sort of movement throughout the body. They may not have true muscle tissue, but all of them are going to be able to use that nervous system that they have, even if it's a very primitive nervous system, to react to their environment and move around their environment. And then, of course, as we get farther along sort of in our travels to ending up with mammals, we start to get more and more developed nervous systems, more and more developed muscle systems, and those are going to be increasingly complex and coordinated. All of our organisms are going to go through some level of development after reproduction. So most of our animals are going to reproduce sexually, meaning we're going to combine two different genomes. This is going to involve a sperm and an egg, just like in plants. You are probably very familiar with this at this point, having gone through Biology 101. For our animals, most of them are going to go through very specific stages of development. Some of these stages might include very specific forms that are different from the full-grown or fully developed organism. Others are going to be a little bit different. It just kind of depends on the development pathway. For fertilization, some fertilization is going to be internal, like in humans. Some is going to be external, like fish that lay eggs, and then the males that basically layer sperm on top of them. And likewise, embryos are going to develop inside the mother, and some of them develop in the environment. So we'll talk about those different strategies. Develop inside the mother, like our mammals, just like ourselves, and then others in the environment, those that lay their eggs somewhere in the environment. Some organisms are going to undergo metamorphosis. We won't talk too much about metamorphosis, but this is a key feature of our arthropods. Basically, our insects. And this is where the organism has two distinct forms, a juvenile and an adult form. You're probably familiar with butterflies and moths that start out as caterpillars and then produce a chrysalis, and within that chrysalis they undergo this metamorphosis. This is a complete body structure change. This is the same organism. This is not an alternation of generations type thing. This is one organism structure developing into a different type of structure. And then that full-grown insect, in this case a butterfly, comes out of the chrysalis and has this new and distinct form. Here we have the current accepted phylogeny for animals. Just like our other phylogenies, these could change as we get more information. But our later animals in our phylogeny are probably pretty well set. Back here at the beginning, the Cnidophora, Porifera, and Cnidaria. These ones have more recently kind of shifted in their organization. The Cnidophora are the most early diverging. They would have come from a common ancestor in the Choanoflagellates. So in our lecture on protists, we talked about the group called the Choana Monada. And this is a group of organisms that have almost a jellyfish-like structure, but they don't fall into the jellyfish category with the cnidarians. They have a collar around that has a set of tentacles with a flagella out of the bottom, and they are basically filter feeders. So our very first lineage split comes with the Cnidophora separating from an ancient group of choanoflagellates. And then the choanoflagellates give rise to all of these other organisms. There are about 35 recognized animal phyla in this group. We are not going to cover all of the animal phyla. We're not even going to cover all of the animal phyla that are here. But for each one of these groups, you're going to need to know the general characteristics of the group. For some of them, these are very big, broad categories. And you'll need to know like one or two good examples of organisms in there. I would suggest knowing the one that is going to remind you the most of what characteristics these organisms have. Keep in mind that some organisms lose the defining trait of their common ancestor or their shared ancestor with these other groups. And so we may not see all of these traits in every single one of these groups. Some groups are going to lose characteristics, some groups are going to gain characteristics. For the exam, you'll need to fill out an animal phylogeny of invertebrates. So this is our invertebrate phylogeny. And then you'll also need to fill out a phylogeny for the vertebrates. So the vertebrates are going to come out of the chordata group over on this side, and there'll be a separate phylogeny for chapter 27. One of the key characteristics, or two key characteristics that separate our animals, make them monophyletic compared to the other organisms in the Opisthokonta lineage within the Unikonta supergroup, is the origin of multicellularity. And we definitely saw multicellularity in plants, but multicellularity came up multiple times. And then the origin of two germ layers. We'll talk later about germ layers, but they are essentially layers of cells that happen early in development, and then they develop into different tissues within the organisms. Another split we have here are what we would call the metazoa. So zoa means animal. Metazoa are the organisms that don't have true tissues. So these guys have no true tissues. But then everybody else, all of these bilateria over here, are sometimes called the eumetazoa. And these are the organisms that have true tissues. So we'll start to see those true nerves, true muscles, and true tissue layers within these organisms. So there's kind of some big, broad categories here. The metazoa, we'll start with them, then we'll move into the eumetazoa. We'll talk about what all of these different categories are: bilaterians, protostomes, deuterostomes, Lophotrochozoa, and ectozoa, and then of course the chordates. Before we really get into all of these different types of animals, we're going to talk about some of the common characteristics that we're going to see. Most of these characteristics are going to be of our eumetazoa organisms, not so much the metazoa. The common ancestor of all of our animals were known as choanoflagellates. And they come out of the Choanomonada. Choanoflagellates are protists. They are single-celled eukaryotic organisms. And it is thought that groups of these choanoflagellates came together and basically evolved multicellularity by coming together. They are flagellated protists. They have tentacles around the bottom, so they do look a little bit like jellyfish. They're not actually jellyfish, but they do look a little like jellyfish. They've got these tentacles around the bottom, and then they've got the flagella out the back that's going to help them in their aquatic environment. The function of those tentacles is to help these organisms act as filter feeders. So they're going to pull water in through their tentacles into the center of this organism and be able to take in their nutrients that way. Our early animals would have been something similar to these choanocytes. And the reason that we think they were similar to the choanocytes is that there are some characteristics in the cnidivore group, the very earliest diverging group, that are sort of reminiscent of these choanocyte organisms. But also we see choanoflagellate almost identical cell types in our sponges, and we'll talk about those in just a second. But you can see up here we've got the choanoflagellate, that is the independent organism, and then we've got what we call this collar cell. This is called a choanocyte, and these collar cell choanocytes inside sponges are part of a larger multicellular organism. So they're in sponges, not their own individual organism anymore, but they've become part of a multicellular organism. They function in almost the exact same way as the individual choanoflagellates. Got an image here. You can see the tentacles off the back and the flagella, and then we've got a nucleus in the center here. Traditionally, biologists have classified animals based on 3 distinct body plan descriptions. One is body symmetry, the next is germ layers, and then the third is embryological development. So within body symmetry, we basically have asymmetrical, radial, and bilateral. For germ layers, our animals are going to have basically 2 or 3 germ layers, except for our sponges, our poor sponges, which have no germ layers. And that is because they lost those germ layers during evolution, their evolutionary pathway to modern sponges. And then finally, we're going to talk about the embryo development in these different animal types. So symmetry refers to balanced proportions. Usually we talk about a particular plane through which we see symmetry. Our sponges have no plane of symmetry, meaning they essentially have an irregular shape. There are no 2 sides that are the same. No matter which angle you look at them from, they look a little bit different. We might also see radial symmetry. Radial essentially means circular, so circular symmetry. What this means is that no matter where you cut a plane through the organism, they're going to be identical halves. So if we look at this cnidarian here, if we looked at it from the top, we could cut it along this plane and it would be identical on both sides. We could cut it along a different plane and it would be identical on both sides. And we could do this around any plane at any point in this organism. When we talk about symmetry, we may not see the same symmetry in both the larval and the adult forms. But when we talk about our next group, the bilaterians, we're going to see that at any stage in development, they may go through that symmetry. So some of our organisms are going to be radially symmetric at some point in their lifespan and bilateral in others. So the radial symmetry may not stay throughout their entire life, but in general, an organism is going to have no symmetry, radial symmetry, or bilateral symmetry. Most organisms that are bilaterally symmetrical are going to remain bilaterally symmetrical through all of their lifespan. There are a couple who have radial symmetry at certain times in their development, but as long as they have bilateral symmetry at any point in time, they are considered a bilateral organism. So once we pass our cnidarians, once we get to our bilateral protostomes and deuterostomes, all organisms are going to have bilateral symmetry at some point in their development. Bilateral symmetry means that there is a single plane down the middle that makes two halves. And on either side of the half, they should be identical. You can kind of think of this as a mirror image. Down that center plane of symmetry, you've got a mirror image for the body plan. So we've got a couple of examples here. We've got this blue butterfly. If we were to draw a plane of symmetry right down the center, be the same on both sides. We see this in mammals as well as many of our arthropods. Pretty much everything from this point on is going to have bilateral symmetry. And we call this whole group bilaterians. Our bilateral organisms are going to have some specific directions. We don't see all of these same directions in our asymmetrical and our radially symmetrical organisms. As far as the bilateral body plan, we have the dorsal and ventral, or upper and lower sides. So we're going to use the sea turtle as our example, but think about an organism that is crawling, swimming, walking on all fours with its belly towards the ground. The upper side is going to be what we would call the dorsal side. So the way that I remember this one is the dorsal fin on a whale or a shark is that fin that sticks up out of the back on the upper side of the organism out of the water. That's just how I remember that. But dorsal is going to be the top or the back of the organism. Ventral is going to be the lower, so the underbelly side of the organism. And these two sides are going to be a little bit different. So if you just think about your front and back, your ventral and your dorsal, you have different characteristics on the front and the back. They're not identical. Anterior means head. Ante means before, so this is the structure that comes before. So if you think about being on all fours or down on the belly, there's going to be an end that we would call the head end. The head may or may not have a brain. It depends on if it's a chordate, what level of nervous system it has. But usually there's going to be some sensory organisms up in the anterior regions of the organism. And then posterior, post means behind. This is going to be the tail end of the animal. May or may not have a tail, but it's going to be the back end. And of course, the front and the back are not the same either. We've already talked about it, but right and left sides are going to be mirror images of one another. So this blue plane down the center, that is showing our bilateral plane. The most important feature of bilateral symmetry is going to be cephalization, or that anterior head end. Most of our bilateral organisms are going to have highly specialized head structures. And this is going to put those sensory structures at the anterior of the organism. This is going to be important because the front of the organism is going to be the first part of the animal to encounter the environment. So it makes sense then that our sensory structures would be up at the front, the anterior of the organism. So if you think about pretty much any animal that you want to think about, they have a direction that they travel, that they encounter the environment with first. And in most of these organisms, we're going to start to see that that is where there is a centralized nervous system of some sort.